Hello and welcome back to the Biochemistry for Health Sciences channel. In the previous videos, section 5, we had about 30 or so videos where we discussed various aspects of human metabolism. In this section, this video section 6 onwards, we shall look at this very important question, and that is, how is metabolism regulated? Blood flow to an organ is a very important regulator of metabolism. For example, if this cell requires more glucose so that it can generate energy, in that case we have chemicals such as carbon dioxide or acid or even hormones. And these chemicals can cause vasodilation. It can make the blood vessel bigger. And when the blood vessel gets bigger, there is more blood flow. More blood flow means more glucose will be able to enter the cell. And more glucose means the cell will be able to make more ATP. Another important regulator of metabolism is the transport protein. Many chemicals, for example, glucose, cannot simply travel through the plasma membrane. They require a transport protein. For example, glucose requires glucose transport proteins. So the nature of the transport protein, the properties of the transport protein, the number of transport proteins, as well as the function of the transport protein or the location of the transport protein, all of these can be changed. And by changing these, that's going to affect or regulate metabolism. Since most of these reactions that we see in metabolism, these biochemical reactions, are catalyzed by enzymes, therefore it's not surprising that one of the best ways to regulate metabolism is by changing either the function of the enzyme, making the enzyme more active or less active, as well as by changing the number of enzyme molecules, either increasing the number of enzyme molecules in the cell or decreasing the number of enzyme molecules in the cell. So let's look at a few examples of how metabolism is changed by changing the transport proteins as well as by affecting enzymes. Consider the glucose transport protein. So neurons and red blood cells, neurons have predominantly GLUT1 and GLUT3. These are proteins that transport glucose into the cell. Red blood cells have predominantly GLUT1. When you look at the Km for glucose, these proteins, GLUT1 and GLUT3, they have a very low Km, about 1 millimolar for glucose. That's roughly about 18 milligrams per deciliter. This means that when your blood glucose concentration is somewhere in the normal range, low normal here, 60 milligrams per deciliter, that's about 3.3 millimolar. These proteins, GLUT1 and GLUT3, they will work at 75% of their maximum capacity, 75% Vmax, because they have a very low Km. However, when you look at this glucose transport protein GLUT2 that is found in hepatocytes in the liver or in the beta cells in the pancreas, these proteins GLUT2 have a very high Km for glucose, about 20 millimolar, that's about 360 milligrams per deciliter. So at the same blood glucose concentration of 60 milligrams per deciliter, these proteins work only at 15% of their maximum capacity. So because they have a very high Km, they work at a very low rate. So this would mean that at 60 milligrams per deciliter, most of your blood glucose, the glucose in your blood vessel, will prefer to enter neurons and red blood cells, and will have a harder time entering hepatocytes or the beta cells in the pancreas. As we discussed in starvation, this is very important because when your blood glucose is on the lower side of normal during starvation period, it's very important that the glucose be reserved more for the neurons in the brain as well as for the red blood cells, which rely very heavily on glucose for their energy needs. Another strategy that the body uses to regulate metabolism is the location of the transport proteins. For example, after a meal, when blood insulin levels are high, this glucose transport protein called GLUT4, which is predominant in adipocytes as well as skeletal muscles, this transport protein for glucose is actually found on the plasma membrane of the skeletal muscle cells as well as the plasma membrane of the adipocytes. So after a meal, when glucose levels are high, glucose can actually be taken up by adipocytes as well as skeletal muscle cells. However, during starvation periods, this muscle protein as well as the adipocyte GLUT4, they will go back into the cytoplasm. So during starvation periods, we do not see this GLUT4 on the plasma membrane of adipocytes or skeletal muscles, which means they would not be able to take up the glucose from the bloodstream. And that is wonderful news for the red blood cells as well as the brain. That means the brain and the red blood cells and other cells that require blood glucose during starvation periods when plasma glucose levels are low, they would get the glucose rather than adipocytes or skeletal muscles. They don't have to compete with the skeletal muscles or the adipocytes. So the location of a protein within the cell or on the plasma membrane of the cell is yet another way of regulating metabolism in the human body. Another way to regulate metabolism is changing the number of transport proteins. So if this cell wants to get more of a chemical, if it wants to get more glucose, it can always increase the number of glucose transport proteins, and therefore more glucose will enter the cell. And if it wants less glucose, it can always decrease the number of glucose transport proteins, thereby decreasing the amount of glucose that enters the cell. Changing the function of transport proteins, the activity of these transport proteins, how fast they transport a certain chemical across the membrane, is yet another way to change or to regulate metabolism. For example, there is a transport protein called nucleotide sugar transporter that is found on the Golgi membranes. And this transporter will transport chemicals such as CMP sialic acid across the Golgi membrane. This transport protein is inhibited by methylated CMP. So when there's methylated CMP present, this will affect this transport protein, and the transport of CMP sialic acid across the Golgi membrane will be reduced. The inhibition of transport proteins by drugs is a very important clinical application of this concept. For example, the drug digoxin that is used for heart failure patients, this drug will inhibit this transport protein called sodium potassium ATPase in cardiac cells, and that causes increase in intracellular calcium in the cardiac cells, which will lead to a lower heart rate, but an increased myocardial contractility, which will help promote the heart function. Since metabolism involves many biochemical reactions, and most of these reactions in metabolism are catalyzed by enzymes, so it's not surprising that one of the best ways, one of the most common ways to regulate metabolism is by either changing the enzyme function, the catalytic activity of existing enzymes, or by changing the number of enzyme molecules, the amount of enzyme molecules. So the cell could increase the enzyme function or decrease the enzyme function, or it could increase the number of enzyme molecules, or it could decrease the number of enzyme molecules in order to regulate metabolism. So in future videos, we will see how the cell changes the catalytic activity of existing enzymes by various strategies. For example, sequestering enzymes in compartments, changing the availability of cofactors, allosteric regulation, covalent modification, conversion of an inactive enzyme or zymogen into active enzymes, and then the use of isoenzymes. And then we'll discuss the strategy of changing the amount of enzyme, the concentration of enzymes in the cell. That can be done by changing the synthesis of new enzyme molecules, increasing or decreasing the synthesis, or increasing or decreasing the degradation of existing enzyme molecules. So between these two processes, we can either increase the amount of enzyme in the cell, or we can decrease the amount of enzyme in the cell. So we'll discuss this in future videos. So until next time, thank you once again for watching. Goodbye and have a wonderful day.